you very much for this massive embarrassment of an introduction. One thing that I thought I was going to do when I retire many years from now was to write my biography, but I just see, as you saw, that it's been done. Thank you, the IMP community, for asking me to join in honoring the memory and the leadership of Max Birnstiel, the founding father or the founding director, perhaps accompanied by other founders, of this great beacon of science in the heart of Europe, the IMP, which from the very beginning was a tremendous presence and is a tremendous presence of science in Europe, biomedical sciences, and inspiration for many institutes of this kind. The inspiration for institutes and communities that were asking themselves, is it possible to create a new entity, a new endeavor of excellence in a Europe that has, of course, centuries-old universities and institutes, very wealthy ones, very large ones? Is it possible? And I was part of a community asking this question exactly 20 years ago. Is it possible in 2002 to place in Barcelona something aside from the universities to help and complement what the universities have to provide as a venue for cutting-edge science? And the example that we, my colleagues, in founding the IRB, the Institute for Research in Biomedicine in Barcelona, the example that we were turning our heads to, and we were turning the politicians' heads to, was you, the IMP. They've done it. If they've done it, it is possible if you orchestrate this well. So you have had an impact well beyond yourself and the science that you produce, encouraging others to enable themselves to place more entities of your kind on the European map. I'm extremely proud and grateful for having been able to contribute to European science through that particular intervention. And again, a great honor to be here today and to add to the roster of those who have preceded me as speakers in this distinguished seminar series, lecture series, in front of this August assembly. For students and for everybody really, but especially for students and postdocs, I like to place my lecture in the context under the overtones of how, at least in my case, as Anna referred, science can be pursued in a way that is effective within one's limitations and capabilities, and it's fun at the same time, and it counts, which is let your work be mission-inspired but curiosity-driven. Within a mission, follow your curiosity, nothing else. That has worked well for me, and today I want to share with you some perspective on a problem that is related, is in fact front and center in oncology today, that of metastasis, the source or the reason, the cause of 90% of cancer deaths. Why is metastasis so difficult to cure? 90% of deaths from cancer are due to it. Well, we know that metastatic cells are very heterogeneous. Tumor heterogeneity, it's a phrase, tumor heterogeneity, and there is the notion that this must be even worse in metastasis. We'll see about that. Regenerate a tumor from a single cell can form a tumor out there and kill someone. They can adapt to different organs and their ecosystems, and they can rapidly develop resistance to therapy. The metastasis steps have been delineated, drawn in cartoons, analyzed, they are understood. Dispersion phase that involves cellular functions that have been the subject individually of whole symposia and books, migration and invasiveness and so forth, circulation, how they circulate and all of that, fine, look, they distribute to the four winds. Then they infiltrate distant organs. The vast majority that try die, but those that do don't go on to form a metastasis right away. They pass through a period that can last from months to decades, literally, of a so-called dormancy, dormant metastasis, the segment of the entire process about which we know the least. We know more about the overt metastasis state, because there is a lot of it, takes place in different organs, in commingling with the host cells in that organ, and that can be studied and described with great profusion of details, as we and others have done, as an example of adaptation first and co-option later. But we must understand metastasis not as the deed of some malignant cells that go out there and crawl and interact and maybe avoid immunity. It's really an interface between these cells and the entire body ecosystem, certainly the entire local tissue systems that are very different in the barriers to metastatic outgrowth and seeding and outgrowth that they present, but also with that in the opportunities that they present. You know, metastatic growth in the bone marrow is very different and resorts to very different sources of support and have to fend off very different sources of hostility from the same in the brain or in the lung or in the liver. That goes without saying. But it goes beyond that. Metastasis is not just, and cancer in general, not just a tumor microenvironment, it's a body macro everything deal. When one cancer and metastasis something happens in a person, not in a cell that we draw in our cartoons. And that person can tell you everything that is going on around that tumor, and how, as researchers, we are understanding more and more, we are beginning to enter the ability to understand more and more, better and better, how these other systemic components affect not only the growth, but the chance that there will be a relapse to begin with, how this will react to a therapy, how the immune system will finish off the job, perhaps with a little help from immunotherapy or not, etc. This is how we feel, my colleagues and I, and many in the world today, that metastasis, cancer in general, must be investigated. With that, we are changing paradigms in terms of therapy and our perception of how the solutions will come. The last quarter of the 20th century and up to today, really, there has been tremendous emphasis on identifying the basis for cancer through genes that are mutant, genes that are defective, sequence tumors, get the genome, study mutant genes in the context of cancer genomes in order to identify gene products that could be targeted with a designed therapy, what we call precision medicine in the form of targeted therapy. That has been fantastically revolutionary at the time when there was nothing that could rationally be devised against cancer, but we soon saw its limits. All kinds of forms of resistance, the fact that many tumors do not have actionable mutations, and so forth. For metastases, that has been even a more limiting approach, because as one scrutinizes more and more, larger and larger, deeper and deeper data sets of mutations associated with metastatic disease, in search for metastasis-causing mutations, it is a very dry well. Metastasis, by and large, is not driven by genetic events, it's driven by epigenetic or postgenetic ecosystem events. This is extracted from a recent paper with my colleagues at Sloan Kettering, led by Nicholas Schultz, in which, leveraging the massive sequencing of all tumors from all patients at Sloan Kettering these days, of admittedly just a limited set of 500 or so genes, but looking at 25,000 patients, nearly 100,000 tumors, a wealth of information about association of mutation patterns in genes of interest with tumor behavior, 21 metastatic sites, etc., etc. Great resource, but one of the upshots is that the specific mutations do very little to explain how and where individual cancers spread. I'm sure that there will be mutations that will specifically drive metastasis, but a very hard look for search for them has turned up really none so far. There are mutations of metastasis, but they are there conferring resistance to the therapy, androgen receptor or anti-estrogen receptor therapy, not mutations that make a cell that was malignant tumor in the primary tumor, souped up and to become metastatic. How did we get into that? We got into that by turning a microphone off. Okay. by the quest that Anne already referred to, which was to understand a powerful regulator of metazoan cell biology, the TGF-beta pathway, important from early embryo development, gastrulation, all the way to the adult reparation and homeostasis of our tissues, and involve abundantly in disease, developmental disorders, immune disorders, and cancer. The TGF-beta pathway, we were busy for some time and continue to be very busy going deeper as it relates to metastasis. But it gave us the identification of the receptor components, how these components interacted with each other to formulate a receptor mechanism, how this is transduced to target genes. But as we were confronted with biology that presented very different responses to TGF-beta, depending on the context. And we initially thought that there might be different variations of this pathway, as it happens in other systems. Different receptor subtypes or types, maybe different pathways and so forth. Yeah, there's a little bit of that, but that doesn't explain a lot of the context-dependent role of TGF-beta. The contextual nature of the responses to this one signal had to do with the character of the cell. It's how cells read signals, as opposed to what signals tell cells to do. Of course, it's a combination of the two. But there were context-dependent transcription factor and epigenetic setups of target genes that determined to the largest extent what the response was going to be. And we had to confront that and embrace that when facing specifically the biology of TGF-beta as it relates to cancer. TGF-beta is a known and powerful enforcer of homeostasis in the immune system, the most potent and broad-acting immunosuppressive or immunoregulatory, immunotaming signal, as well as epithelial homeostasis, making sure that there is the right amount of daily replenishment of the tissue, not too much, not too little. And then tied to that is a very potent tumor suppressor. In pancreatic cancer, half of the tumors have lost SMAD4 or TGF-beta receptors, smaller proportions in other cancers. But then already in the 90s, these different types of activities were compounded by the increasing realization, late 90s, when we began to notice that TGF-beta is actually pro-metastatic, that cells that have graduated from the early primary tumor stage will be very happy to retain adulterated but nonetheless active responsiveness to TGF-beta for better dissemination, EMT, invasion, colonization, and as we have learned more recently, also to put themselves into immunoevasive dormancy. How does that happen? Well, the answers to that came after a lot of scratching our heads and thinking not so much as signal transduction transcription biologies, but as biologies of the cell, of the character of the cell. Early on, and that was the easier part, the cell cycle inhibitory action of TGF-beta could be explained by cloning, identifying some of the CDK inhibitors. In fact, my first talk at the IMP was in '94, invited by then budding leader Kim Naismith. Soon after, the lab isolated, purified, sequenced, and cloned P27 KEAP1 CDK inhibitor. CDK inhibitors get mobilized in response to TGF-beta, and the transcription factors that enable that are FOXO transcription factors, and that gives you cell cycle arrest, and that's all understood. But this happens in normal epithelial cells in the context of homeostasis. Then something happens that when these cells are transformed by typically RAS mutations, as initiating mutation or in the initiating pack, then something happens. And that is something that transforms these cells in the response to TGF-beta from just a homeostatic modulatory function to a killer, to a tumor suppressive signal that induces apoptosis. And we appreciated only recently that that followed an EMT. TGF-beta is a very powerful, very common inducer of EMT from early development to throughout. It induced a super powerful EMT in these premalignant cells, much more powerful than in their normal counterparts and progenitors, but within 24 hours they were dead of apoptosis. Now, if these cells are going to graduate into carcinoma states, they have to solve this, and they do by any one of two routes, either by accumulating disabling genetic mutations, alterations in the TGF-beta pathway, case of 50% of pancreatic carcinomas, smaller proportions in other carcinomas. Still, at that stage, they are still happy to have TGF-beta around because it's a very powerful immunosuppressor. It also acts on cancer-associated fibroblasts to create intratumoral fibrosis that is supportive for tumor growth. The other route, more commonly embraced by premalignant cells graduating past that bottleneck, is by decoupling TGF-beta signaling from apoptosis. And we found a number of ways in which that happens. With that, they are even better. Not only do they have TGF-beta action on the environment, supporting them, but they can respond to TGF-beta with EMT and the production of cytokines and other factors that allow for a better interaction with the host environment, as in the context in which we realize all these things. Bone metastasis, TGF-beta action on breast carcinoma cells produces factors that mobilize osteoclasts for osteolytic metastasis. The thing that was harder to crack, however, mechanistically, was that step in the middle. Then something happens. What was that? Why is it that hyperactive RAS signaling leads to apoptosis? The answer for that came a couple of years ago when Jessie Sue did a screen to search short hairpin still in those days screen to look for factors that would enable the RAS pathway to collaborate with TGF-beta signaling in inducing a very strong EMT. And she found that there was a factor that, in spite of its lofty name, RAS response element binding protein 1, was almost completely forgotten in the RAS field and in the field at large. RED1 is indeed the MAP kinase activated factor that then cooperates with SMAD, the contextual RAS mutational RAS context factor that collaborates with SMAD in organizing a very powerful inducer of EMT transcription factor signaling that leads to EMT. However, this is happening with TGF-beta still trying through non-rep dependent but SMAD-dependent signaling processes to impose an epithelial fate on these cells. So these cells are getting B epithelial, B mesenchymal. At the same time, is one of those conflict signal states in biology that the cell says, you know what, I'm out of here, apoptosis. Now, we went on to find that, on the one hand, how to decouple that signal from apoptosis or EMT-inducing signal from the apoptotic liability, with details I will not get into, but the papers are listed here. And when that happens, then a number of other factors that are expressed together with a SNEL EMT transcription factor as part of an EMT that is meant to be regenerative, that is meant to also incite fibrogenesis for extracellular matrix production in the context of a tumor, very supportive of tumor growth. That is when that happens. It's a fibrogenic EMT where both the SNEL and fibrogenic factors, interleukin-11, PDGF, and others, are all co-regulated by a SMAD-Rep1 to form what physiology was meant to put under TGF-beta RAS signaling, in which is reparative epithelialization growth. In the context of tumors, however, this is a major engine for metastasis. We knock out Rep1, and lung metastasis, as you see here, practically evaporates. So this is the kind of education that we have been accumulating, what has forced us to think in different ways about the nature of responses to cytokines and what we mean by context when we talk about biology. So with that, what about metastatic progression? Well, the three phases that I mentioned and that you all know are here. This is metastasis as one looks at it from the standpoint of the patient and their oncologist, is something that may well be seeded already by the time a tumor is diagnosed and removed and treated. That seeding stays dormant for a period of time unless it was already actively growing, in which case that's a case of diagnosis of stage 4 carcinoma. But more and more these days, carcinomas are detected at early stage 1, 2, 3, and then one hopes for the best. Applies adjuvant therapies of various kinds and hopes for the best. And sometimes the best doesn't happen. And when that doesn't happen, there is metastasis outbreak. Treatment is effective at debulking more and more, but rarely at curing. There is always now a more robust remnant that will engender a new round of relapse. So that is the problem. And from the cell biology standpoint, we draw these cells like these to parallel this human personal, drama. Cells that disseminate, and we've studied that a lot, as I said. Cells that establish themselves in dormancy, that have been thought of cells being just dormant, quiescent, doing nothing, and then something happens. It was sunny in Vienna on Thursday, so they wake up, and then we have trouble. We'll see about that. And then that waking up takes, of course, a very organ-specific character, as this expanding colony has to specialize in making the best out of that environment. We started by the latter in the lab, by studying the latter, intrigued by what TGA-beta was teaching us, but now wanting to take metastases head-on. And we reasoned that if in a patient cells from a common tumor, a breast tumor in this case, were metastatic to multiple organs, if we would have a sample, for example, what accumulates in the pleural fluid of these patients, we would have representative of the multi-organ demographics of metastatic cells coming out of that body already adapted, having been adapted to one or the other organ, using the mouse as a cell sorter, isolate different metastatic cells from different sites, verifying that yes, they had that preference for one or the other site, and then doing these various steps that led to many stories, many findings for multiple tumor types, breast cancer and lung adenocarcinoma especially, but also renal carcinoma, colorectal cancer, metastasis to multiple sites as listed on the left. And then the genes that were associated with this metastatic character explain specific steps, extravasation steps in those organs whose capillary walls are tight, non-permissive, seeding, coping with the shock of the new environment, that is where they seem to play a role, and colonization, factors that are an advantage, a plus for metastasis, because at that site they can intervene in a way that is very specific and gives any cell that happens to be expressing these genes an extra advantage. Lots of work, very productive papers and careers and so forth, but that is the essence of it. You can find genes that mediate fascinating biology. Some of these genes provide answers to and how does TGA-beta further augment metastatic outgrowth once it has helped colonization. And so there are answers there. Fascinating stories of cancer and tumor cell biology. For example, work of Manuel Valiente first and Qing Chen and Adrien Bois later on the interface between brain metastatic cells and the astrocytes, the most abundant cell in the brain, or one of them certainly. How initially the astrocytes are out to kill the intruders by production of killer cytokines, Fas ligand, and how brain metastatic cells have devised an antidote against these inhibitors of the protease that releases Fas ligand. How later, as they have installed themselves and fend off the hostility of the astrocytes, they are happy to have close interactions with them by establishing gap junctions through upregulation of PCDH7 and connexin 43, through which things like cGAMP, the mediator of the STING pathway, flow to activate in the astrocyte, making the fall guy that will produce cytokines that in that context are salutary, levels of interferon, type 1 interferon, and TNF that are supportive for the proliferation and chemoresistance of the cancer cell. This is what was found. These are fascinating cases of the biology of metastases, give ideas about clinical trials, and Adrien has completed and written up a clinical trial on using a gap junction negative modulator to address brain metastases in advanced cancer patients. and so forth. How effective these therapies can be that target a particular component of metastasis to a particular type with or no, I'm sure they can be helpful in certain contexts, but we really want to focus on what will be common to all sites and hopefully multiple diseases. Nonetheless, mission-inspired but curiosity-driven, we were curious about what you see on the left, what the cancer cell in green is doing after having infiltrated the brain. This cell came out from the purple capillaries, and instead of sort of walking away and being perhaps near capillaries for oxygen and so forth, but that's it, now it sticks to the capillaries and it stretches over them. And that was remarkable. And that was just not one vignette. This is what we see all the time. Here's a result of having divided already twice or three times, 3 cell divisions, 8 cancer cells, forming a sheath. They hug the capillary. You see these little arms here hugging and spreading on the capillaries. Well, so we went on to explore this further. He is looking with light sheet microscopy using cleared tissue with a technique called iDisco that zooms into the brain and sees now in magenta the cancer cells, how they place themselves in the capillaries, having been injected into the circulation of the mouse, arrive in the brain and extravasated. And they are there, a couple cells, as you see here, to the left, moving to the right, to a sheath that engulfs the entire local network, and then in cyan, the microglia aggregate reacting to all of that. This is the power of imaging for these things. But the point is that there is a remarkable form of initiation of colonization in the brain, and as it turns out, in other sites, where these cells, which first extravasate, migrate up and down the vessels, looking for spots, we don't know what that is, pushing out of the way pericytes, that's how tightly they can embrace, and then hug, stay put, and start to divide. We identified the molecule that allows cells to spread and adhere in this manner is L1CAM, a cell adhesion molecule that establishes interactions with itself for cell-to-cell homophilic interactions, as well as interactions together with integrins with laminin for integrin/L1CAM co-signaling. Embra-Earth worked out the signaling pathway in at least this context of the cancer cell attached to the capillary. That pathway ends up activating YAP and MRTF transcription factors, and that is the green light for the cell to grow. This cell is in the vessel, it's getting nutrients, oxygen, it's cancer cell, malignant, has RAS mutations, still needs that permission in that context to grow. If L1CAM is knocked out, the cells can still circulate, extravasate, stay attached, tethered to the capillaries, integrins we suppose, but they will not proliferate. And in the center of the slide, this is a bioluminescence imaging of a brain ex vivo. You see the 100-fold difference in colonization powers of cells that have or lack L1CAM. This exemplified in the brain most clearly because of the beauty, the elegance of the capillaries in that organ. We later appreciated, knowing what we were looking for, that is general, that it's in all organs that we looked at and by most, not all, carcinoma models that we examined. So that is interesting because it gives us the first example of a mediator of metastatic colonization that seems to be pretty important, if not essential. That is for multiple types and multiple organ sites. So we became super interested in this one as a potential target. This is the work showing why L1CAM is appreciated, the signaling process that I mentioned before. But the next question is, if these cells already arrive equipped with the ability to express and use L1CAM, and that makes them very effective metastasis-initiating cells, colonization-initiating cells, where do they get that from? They arrive there, so they must be coming with it from the primary tumors. Indeed, L1CAM discovered in the early 90s as a cell adhesion molecule very active in the brain. In fact, it's the gene mutated in the L1 syndrome of mental retardation due to very defective development. It had been noted over and over and over, dozens of papers, that is expressed, they said, ectopically, they said, in tumors. And the more there is of that, the worse the prognosis. One of those things that simmers in the literature, and because it's a cell adhesion molecule, must have to do with invasion, metastasis. Yeah, but what? Now we know. But we refer to that information in trying to think about the question, where does this come from? And this is the work of Karuna Ganesh, physician scientist who joined the lab, did the postdoc with us, and is now a faculty member in her own lab at MSK. And she took on as a GI oncologist to look for L1CAM expression in normal tissue and in primary tumors, to see that, as others had reported, the normal tissue, normal colon, does not express L1CAM at all. However, in primary tumors, there is expression of that, concentrated in the invasion front, and there is more of it in metastatic lesions. Moreover, these cells appear to be chemoresistant. After neoadjuvant therapy, metastatic lesions and residual disease, even in the primary site, becomes further enriched for L1CAM expressing cells. What's that? We began to wonder whether this might have to do with regeneration for a number of reasons. To do that experiment, we obtained conditional allele, mouse with a conditional allele of L1CAM, crossed it to a Villen-Cree, also LGR5-Cree. Both of them gave the same result, even LGR5, which is less penetrant, which was to show that in normal mice, like in humans, the normal epithelium is not expressing L1CAM unless you damage it, unless you impose injury. Then there is expression of L1CAM, and this L1CAM expression is retained by the cells even after they have already replenished, just replenished, the epithelium with their progeny as cells of the repair of the damage, using here dextran sulfonate sodium to induce ulcers. In the absence of L1CAM, mouse development, intestinal function is just fine, unless and until you damage it. Then there is no regeneration in the mice. Many mice die of intestinal ulceration. So L1CAM is something that is, or cells with the capacity to express it, are there latent, ready to respond to the sensing of barrier tissue disruption. Then they manifest themselves, and they need that there in order to do their job. And then they will disappear again. So it's for a period of covering, patching the broken barrier that this is important. And indeed, we went on to find, Karuna, that L1CAM is essential for every form of regeneration of tissue, in this case, the tissue being tumor. From patients that she treats, she obtained metastases as well as primary tumor that have cells from organoids, isolated L1CAM high and low cells from these organoids. They overlap poorly with LGR5 cells. It's the L1CAM alone or double with LGR5, not the LGR5 without L1CAM that generate organoids and that transplant tumor. So the L1CAM expressing cells is the carrier of tumor regenerative powers, which is what metastasis and relapse are all about. So that led to this working hypothesis, which distinguishes, and this is no surprise for some of you, but many in the field oversimplify things, and that homeostasis and injury repair are in the hands of distinct forms of regenerative progenitors. Call them stem cells, transient amplify, whatever you want. That's the job they do. They are distinct. They are responding to different moments in the biology of that normal tissue. In cancer, sure, homeostatic stem cells can be and are certainly colorectal cancers the source cell, the cancer stem cell for tumor initiation. But they are not, as we find, as this work suggests, also work of Jacob van Rienen, of a different approach, completely different, but pointing at the same conclusion. They are not the ones that reestablish the tumor. That's in the hands of regenerative progenitors that have been provoked to emerge by the tumor self-disrupting itself, mobilizing the L1CAM phenotype which is the one that is needed at a distance and perpetuated. The fact that this is not a simile, a co-option of something normal, but is something normal in the hands of malignant cells, is something normal, regenerative biology in the hands of malignant cells, is further exemplified by single-cell analysis of tumors. In this case, we are switching back to lung adenocarcinoma. Work of Ashley Loughney, in parallel with Karuna's work, took on to define the cellularity at the transcriptional level of human lung adenocarcinomas, metastases, primary tumors, and normal tissue, to find that among the cancer cells, which are in these tumors a minority, there is great heterogeneity. Tumor heterogeneity, that is so difficult. No, a lot of that heterogeneity is the heterogeneity you can expect produced by a lineage of progenitors and their descendants along a developmental regenerative axis that has been very well described and studied by the leaders of the field of lung epithelial regeneration in this case. When Ashley looked at single-cell analysis data and computationally ordered the single cells by a lung development signature, they all ordered it themselves very nicely. And when she dialed in specific transcription factors, master regulators as they are called, or the various stages of that development, well defined by developmental biologists and regenerative biologists again, these cells beautifully ordered themselves. Great heterogeneity, but it's coming from normal. It's coming from normal. With it, by the way, other interesting aspects of heterogeneity emerge. For example, if one looks at immune mediators or mediators of cell susceptibility to the immune system, what determines that the immune system will recognize that cell as sick, MHC class 1 molecules presenting tumor antigens, natural killer cell receptor ligands, NK G2D ligands, one finds also that they track, very heterogeneous, but they track with the developmental stages. If you want to debate whether NK cells or T cells or this or that are the most important in confronting this tumor, they will be the most important for a subset of the population that developmentally, at least in part developmentally, has its immune liability around those molecules and not the others. How could we prevent relapse? That is the name of the game. That is the inspired mission that we are pursuing. We decided that really the greatest potential is in the least well-known segment of the pathway, dormancy. What do we know about these cells and their life and how they see it and what they do, what wakes them up, etc. Because if that happens, that is where the deal comes down. If that population awakes, that is where metastasis will emerge, and then you have all that organ-specific adaptation, or all that wonderful biology with you publish papers. You publish more papers than you cure patients, perhaps. So what if we can learn more about things that are general, like L1-CAM, that pertain to the initiation of metastatic colonization by these cells? For that, we had to develop models, and Srinivas Maladi a number of years ago did that by resorting not to later stage cell lines derived from patients with later stage cancer, but cells derived from patients with early stage lung or breast carcinomas, also from mouse models, and select for cells that had the phenotype of interest, which was the ability to install themselves in different organs, multiple organs in fact, and sit there without doing much, although on occasion one of those 10 or 20 mice will come down with a lethal metastasis, like in patient populations. The properties of these cells were one seen is like, yeah, sure, we could have expected that. But we didn't. We had to do experiments. They occupy perivascular niches. They express early progenitor markers, SOX2 in the case of the lung adenocarcinoma model. A few slides ago, SOX2, remember, is an early progenitor state gene here in the foregut segment. They are competent to enter quiescence, self-driven quiescence. They don't enter quiescence because some horrible suppressive force in the environment, TGF-beta, maybe made them do that. Sure, they have hypersensitive responsiveness to TGF-beta induced quiescence, but they produce DKK1 and I'm sure other kinds of Antimitogens, DKK1 is a Wnt inhibitor, Wnt ligand blocker. They are immunoevasive while they are quiescent. They express low levels of NK ligands, and they exist in proliferative clusters. In the models, about 10% of the cells are in these little clusters. They are proliferative. We never see them pass, 15, 20 cells, about 4 cell divisions. If we look 2 months later at mice in the same cohort that were not examined earlier, this is what we see again. So it's not that the clusters that must have been there in this mouse 2 months ago kept on growing. They disappeared. New ones emerged and disappeared. That is what gave us the clue, the eureka moment of saying, metastasis is not just, dormant metastasis is not just cells sleeping, sitting there, waiting to wake up. They are constantly trying. Whatever, sustains the fluctuation, the sensing of disruption, signals, it's constantly enticing a subpopulation to enter a cycle. And they try. And when they try, they get killed. They get killed because at that point they upregulate ligands for immune cells, and that takes care of it. And the demonstration is by taking, you're looking at it while I'm speaking, in mice that are harboring disseminated cells of this model of latency, dormancy that we develop, you deplete them of NK cells, and metastases emerge very quickly in multiple organs, many metastases. I say NK cells, not because it's only NK cells, but because these are human cells in athymic mice. They don't have mature B or T cells. Compared to highly aggressive human cells, these cells, upon depletion, liberation from NK pressure, they are pretty strong. They are also very strong in NSG mice as metastasis growers. Moreover, back to these unleashing a developmental, tracking with a developmental process, single-cell analysis of these populations shows that the dormant cells are mostly SOX2 expressing early progenitors, as are the incipient, these spontaneous incipient growths. But the outbreaks are dominated more differentiated progeny as defined by SOX2. Unless we deplete of NK cells, then everybody can grow. And now we see also plenty of SOX2 cells in those tumors, which is what led us to realize that the SOX2 early progenitor state is especially susceptible to clearance by immune cells, at least by NK cells in this context. And this is how differences in progenitor state-associated immune susceptibility play out to shape a metastatic outbreak. We've now developed similar models that are immunocompetent in an for analysis in a syngeneic mouse. They are derived from the KP, KRAS, P53 lung adenocarcinoma model. We derived these cells from very early stage adenoma stage lesions. We call them KPAT1, KP adenoma stage line 1. And for comparison here, we compare them with cells derived in Tyler Jack's lab, these KP48-2T1 cells. These ones are very aggressive. The KPAT are indolent, depends on how many cells you inject. The more cells you inject, the higher the probability that one will take off soon. So you see that. But if you inject fewer cells, the mice can go for months without an outbreak unless you deplete immunity. And now the depletion can be of all components, and all components as shown in the graph on the right, and CD4, CD8 effector cells, as well as NK cells, each one individually accelerates depleting, accelerates metastatic outbreak. So that gave us an idea to begin to answer the question, exploiting this model of what limits the ability of metastatic outbreaks, even when they happen, from prospering. We can use this model for photo screens to look for molecules that are interfering with this blockade, whereby knocking them out or down will accelerate metastasis, a powerful positive selection in that screen. To answer the question, how are dormant cells cleared by immunity upon entering the cell cycle in the first project of this kind, that the lab now is excited about applying more to other aspects of biology. But the immune system, which are the candidate components that are most important at this juncture. Cells in the indolent state, non-manifest in the form of metastasis, trying and failing because these components lead them into trouble. This is the scheme of how Jinhu with Francisco Sanchez Rivera, Francisco of the Low Lab, designed the guides and the approach to the screen, and how the screen was done. The guide library for a CRISPR screen was transduced into the cells, either model, the human immunocompromised or the mouse immunocompetent host models, and then harvest tumors, metastases that showed up promptly, earlier than expected, likely to contain cells that have been knocked out for an impediment. The libraries that were designed to address the well-known candidates, NK ligands, MHC class I molecules, DNA sensing STING pathway, RNA and LPS sensing pathways, etc. You're looking at the results of the screen. Every pie reports on metastases to 5 different sites. These are the 2 screens in the human and in the mouse cell line model in mice. And then the extent of the black segment of the pie is the proportion of metastases, of outbreaks in that organ by every library. So most of these score to some extent, in some organs better than in others. MHC class I molecules, NK ligands, it's welcome information. They were not a shock to hear. The one that got us intrigued was this one, the STING pathway and its client pathway, interferon. Looking at the individual genes in both systems, the human model, cell line model, and the mouse cell line model, multiple components of those 2 consecutive pathways screen. In other cases, we're just in one model or the other. The STING pathway. The STING pathway, it senses double-stranded DNA in the cytosol, in the cytoplasm. This is commonly present in cancer cells as a result of chromosome instability, microsatellites, and so forth. A very powerful then inducer of these cells' exposure to immune killing because it makes these cells produce interferons and other cytokines. Indeed, when we look now by in situ analysis, we saw that in individual resting cells, the expression of STING is very low. However, when these cells are in Ki67 positive little clusters, they have committed themselves to entering the cell cycle. Now they express STING. And in larger outbreaks, there is still expression of a STING, but less, more isolated cells, and overall is dimmed. So a pattern that was consistent with the STING making trouble for them. The STING targets, CCL5 chosen here, track with that, a marker of a STING pathway activity. And this again is true for both models. In the mouse single cell analysis of the models that I described to you before, that provided an additional layer of information. It is the SOX2 early stage progenitors, the ones that express the high levels of STING. As the population grows and becomes more bulky and more SOX9-like, that is less rich in STING levels, and that's quantitated on the right. Sufficiency and necessity experiments showed that overexpression of a STING, knockdown of a STING in indolent latent metastasis competent cells allows them to form outbreaks more easily. They get themselves less into trouble. In the aggressive models, overexpression of a STING, they express low levels to begin with. Overexpression of a STING gets them into trouble. And this is true both in the human cell line models and in the mouse cell line models. When STING is overexpressed, the tumors that form, in this case by the aggressive metastatic cells, are better infiltrated by lymphocytes and also knocking down specific lymphocyte components, NK cells or effector T cells, prevented overexpression of a STING from giving these cells trouble. This is then corroborated, paralleled by observations in human patient-derived samples. Metastases are generally expressing less STING, although there are some cells that still express it at high levels compared to matched primary tumors. Going to the single-cell analysis, heterogeneity again, early progenitors, SOX2 progenitors, SOX2 high progenitors have the components of the STING pathway at higher level and a signature of STING targets at the bottom, scoring at the higher level than in later state progenitors and progeny. So this is the schematic of what comes out from this first analysis of liabilities that are specific to the cells that are trying to emerge from the dormant state and engage in express L1CAM and do all those things that will make them a macrometastasis. STING is low in dormant disseminated metastatic cells. It reaches its highest, its zenith in metastasis progenitors that re-enter the cell cycle, and that triggers NK as well as CD8-mediated trouble, clearance of the cells. And we are excited, and we are doing these experiments now, to use STING-activating agents, drugs, to get these populations into more trouble, to effectively deplete a body of a mouse, for the time being, of disseminated dormant cells, by getting that population more exposed to clearance by the immune system. And the results are extremely, extremely promising. So to conclude, we are trying to generate the knowledge necessary to prevent relapse by understanding what the various phases of relapse are made of, developmentally, regeneratively, by realizing that this is regenerative stem cells that happen to be malignant, and that they use the devices of quiescence competence, immunosuppressive, immune-evasive quiescence versus immune-exposed proliferation, special interactions with the environment, with the basement membrane, extracellular matrix, with L1CAM, and I'm sure other mediators for that moment in the early stage of regeneration, which is about reepithelializing, building a gap for the broken barrier, again in the hands of cancer cells now. That is the initiation of relapse. Relapse, not only the first time they try by disseminated cells that came from a tumor, but we suspect that it's also what goes on when an overt metastasis has been pushed back very efficiently with therapy, but not 100%. What's left is not just a little bit of what we had before, but a very different little bit, and we suspect that it is akin to this little bit. And with that, I want to acknowledge those who contributed to the work that I presented today. Of course, the lab has many, many other former members, one of which is a distinguished faculty member among your faculty. But this is the work that this more recent crew has presented. I thank you all for your attention and for your welcome. Thank you.